To Mónica Carrasco. She was a student from this university. Now she's working in University of Talca. She's also associate of the Department of Molecular and Cellular Biology of Harvard University. So it's a big pleasure for us to have her here and to hear what she has to tell us about neurodegenerative diseases. So give applause. Applause? Applause?
Thank you to the organizers. Honor to be here. I feel a bit silly because I have to do the talk in English, so please questions, Spanish, English, Spanish too, a little bit of Argentinian, but I think my Argentinian is a bit like, Portuguese, no. No, but what I have to play here. Today we are going to talk about mechanism of neurodegenerative diseases, in special ALS, because that's the one that I'm working currently. But I want to talk to you about something. In terms of neurodegenerative disease, we know that there are many, a broad range, but we have that the most important that affect us as Chileans are Alzheimer's disease, Parkinson's disease, Huntington disease, and lately ALS. The studies about how many Chileans have each disease are very weak, in the sense that we don't have a good catalog of the people who has each disease in our country. That's something we need to look forward, because we need to know these numbers, especially we need to know these numbers in terms of our regionality, because we are such a long country. Certain regions have more diseases on one aspect than others. And this could be a clue to us to understand, for example, what are the environmental cues that may trigger one disease or the other. In terms of neurodegenerative disease, what I would like to understand, and I do not understand, is what is happening. Why is the neurons are dying? And I understand they all die. Actually, in many of the diseases, we have 3 branches of research that shows how neurons die by triggering apoptosis, by triggering some sort of necrosis, or by triggering neurodegenerative changes that leads to cell death. The other thing that I would like to know is when it starts, why this particular neuron needs to die in our system. And the third question, do we have anything in common? Because once the disease are started, independently of how or which disease we are talking about, we always have problems in terms of loss of neuropila, changes in the oxidative stress in the cells. We have aggregations all over the board. We have aggregations in Parkinson, in Alzheimer, in ELA, in Huntington. And from all of these diseases, the only one that we can accurately catalog is Huntington. And this is a problem, because we need to understand what type of disease we are trying to characterize to focus on a type of question. That's one of the main problems of studying in general neurodegenerative diseases. And today we are going to go back to the beginning and then to the aging process. I'm going to show a lot about stem cells. And so I wanted to remind you that when we talk about stem cells, this is a term that has been trashed in the last decade, I think. And what we need to understand is that when we are talking about stem cells, we are talking about cells that can come from different parts, depending on the stem cell you are talking about, but mainly they have the properties to self-renew or to differentiate. When they do self-renewal, they can produce a population of stem cells that is similar. And then, according to environmental cues, they can differentiate and generate different cells in the nervous system. We're going to focus now towards the nervous system. Now, when we talk about the stem cells, we have 3 different properties of the stem cells, 3 different kinds of stem cells, let's say. One that is totipotent, which means that that cell can generate a whole person. The other, it's pluripotent. That means that that stem cell can generate each and every single cell that you have in your body, which is pretty cool, yeah? And the other one that is multipotent, that is kind of pre-programmed to a particular area on your body. And this is important because we are going to talk later on the talk of my experimental model, which is nowadays, since 2006, so we have 10 years of this already, we can take skin biopsies of any of you in the audience, and we can then treat this skin biopsy with a set of virus that are here, and this set of these modifications will induce that this fibroblast, which is a totally differentiated cell, becomes pluripotent. And this is the key word, pluripotent. That means I cannot make a whole person out of them, but I can make any cell that I want. And that's the big difference with these iPS cells and the fetal or embryonic human stem cells, which have been greatly trashed. It has been completely controversial to study them, especially in the United States, where you have several laws and regulations to work with human stem cells that, quite frankly, are very obstructive to do science. Since 2001, with the Bush administrations, that you have a particular restriction to use only 11 lines of human embryonic stem cells. And from those 11 lines, you can only differentiate those stem cells into pancreas. Because the scientists in charge of trying to open the field so that people in the States could work with stem cells were doing research on diabetes. So the idea is that now, with this development from Ichi Yamanaka, we can take any person's cell and transform them into a pluripotent cell. And with that, we have a source to generate any cells in the body. What I wanted to do, of course, is to try to imitate what happened in life. So we know at the beginning we have precursors of neurons. They will migrate to the place that they need to go, and then they will differentiate in a fully mature neuron, and after certain age, which some people say 40, others say 35, which is pretty depressing because it should be 80, but no, it's fairly young on what it means to have a fully function and prime neuron. And after the degeneration happens, of course, we have neuronal death. Now, what we are going to talk today, it's a particular disease. It's called ALS. That's a disease that is a neurodegenerative disease because it kills upper and lower motor neurons. These motor neurons are the one in charge to do a synapse with your muscle and control all of your movements. It's a terrible disease, of course. It's the third most common disease on aging. It's fatal. Usually after onset, people who are diagnosed with ALS live in a period of 3 to 5 years. And death occurs usually when the innervation of the diaphragm fails and persons cannot breathe. And of course, nowadays you know that if you cannot breathe, you can get hooked to a ventilator. Yes, that's common knowledge. The problem with that, and this is something that is hard to see when you don't see the patients, is that not everybody can be connected to a ventilator. Not everyone reacts the same to that type of system. The other thing that happens is that once you are connected to these type of machines, you are very prone to infections. And people end up dying by pneumonia, by sepsia, because the way the system works, it's very debilitating for a patient who doesn't have any control on the muscles. We have been studying this disease for many years, but so far we have actually 2 types of treatments that I'm going to tell you about, and this is the only paper that I have found that tries to catalog the amount or the population of ALS affected Chileans. Here you can see that, I'm going to increase this, that in Chile, if you study the age groups that are affected by ALS, mainly they are affected between 70 and 79 years old. Now, if you see this rate, you can see that it's increasing from the early 90s to this study ends around 2010. So it is a critical condition, and one of the failures we have as a country to try to study this neurodegenerative disease is actually knowing them in terms of the diagnosis. We fail here. We fail big time. Because what happened is that in many cases, people have this neurodegenerative disease, but they are misdiagnosed. And with that, you don't really know how to treat a patient. You don't have that precious window of opportunity that happens when you know somebody's going to get a disease. That happens in Huntington. Huntington is a genetic disorder that causes neurodegeneration, but it's very well known. So people with Huntington can be diagnosed, can even try to do prevention, even though prevention is still in the making. There is this opportunity. We don't have that opportunity for Alzheimer's, for ALS, or Parkinson. And that's what we need to look. First, in Chile, we need to understand how these numbers are now and how do they change in terms of our regions. For example, I don't know if you know, but in the Maule region where I work, we have three main diseases. One is gastric cancer. The second is diabetes, which is kind of pandemic everywhere. And it very strongly on the neurodegenerative disease, we have a population of Huntington patients. We think that this is related. There could be many causes, but we need to figure it out why we have such a concentration of Huntington patients in the region. Now, ALS have early symptoms, including, for example, walking difficulties. Tasks that you do every day and that are very familiar start to fail. Whether it is buttoning up a shirt, tying your shoes. Usually, ALS patients start to see symptoms when at least 60% of the motor neurons have died. So you will figure it out that you have a neurodegenerative disorder pretty late on the disorder. Other symptoms include weakness in your legs, hand weakness and clumsiness, problems with the speech, especially those that have a bulbar onset of the disease will have problems with speech, muscle cramps and twitches. And I remember speaking with one patient and he told me, you know what I felt at the beginning? He said, I feel like I was sitting down and I have a cell phone inside my leg that was vibrating. This is very common on not only ALS, many other neurodegenerative diseases. And these kind of symptoms are perturbing. They are called fasciculations.
How common is that?
Huh?
How common?
Very common in ALS. Very common. The other things are the things that happen at the molecular level. So as scientists, we try to understand what happened at the clinical level, but as molecular biologists, well, I actually was, I'm part of the PhD program of molecular cell biology like 20 years ago, so that's a long time ago. But we try to see what's going on at the molecular level. And these are the proposed pathogenic mechanisms in ALS. And as you read through the list, you are going to figure it out that these mechanisms are also associated to other neurodegenerative disease. Actually, oxidative stress, excitotoxicity. This is the one that I think is the major commonality between all of them. Abnormal protein precipitation and aggregation, cytoskeleton defect, axonal trampor, neuroinflammation, abnormalities in hypoxia, apoptosis, and autophagocytosis. The ones that we could see in ALS, at least in my model, are autophagocytosis, neuroinflammation, cytoskeletal defects, abnormal protein precipitation, excitotoxicity, and oxidative stress. We can check most all of them. And this kind of thing, once the disease triggers, it happens across the board. I mean, it happens on Parkinson-derived patients, Parkinson neurons derived from patients that have the disease, and also in Alzheimer's. The diagnosis, as I told you, is tricky. It has to happen. There are many common symptoms between Parkinson, ALS, dementia, And so what happens is that you go to the neurologist and they have to test you at the level of electromyography. And one of the critical tests is to measure the velocity of conduction. And also they have criteria. One of the criteria, the clinical criteria is this. You're going to have probably the slides later. So I don't want to take too much time on this. And in terms of treatments, there are very few treatments for ALS. One, it's this one, Riluzole, which is the classic treatment that has been used for 30 years now. It only increased the average lifespan of a patient for 3 months. And now, if you remember with the campaign that was called the Ice Bucket Challenge, I don't know if you remember these guys that were throwing up buckets. So ALS Association and other research institutions managed to get a lot of funding to try to accelerate treatment. One of these went into this group that tried this drug that is called edaravone. This drug is now FDA approved in a process that is a fast-track FDA approval. It's super expensive, and it only happened because of the Ice Bucket Challenge. And the idea is that this group, in 2017, generate a 6-month trial that used this drug given in intravenous infusion, typically 10 to 14 days in a row, once a month. And in those 6 months, they saw an increase in those scores that we were talking about just before. So we now have a new treatment. We did have the old treatment, but in general, we don't have a cure. I don't envision to even see the cure of my lifespan, but at least what I will envision is to stop the progression of the disease. Even if we don't find a cure, if we could stop the progression, we are good to go. Here, it's a bit of genetics. We know that in the case of ALS, 80 to 90% are sporadic, 5 to 10 are inherited, and within those 10%, 20% are families that carry a mutation in superoxide dismutase. There are others mutations associated to ALS. Lately, the most relevant one is C9orf72, which is a gene responsible or associated to 30 to 40% of familiar cases and also present in some sporadic cases. I focus my studies on a family or a patient that carries a mutation in TDP43. When we start this research, that was like 2008, 2007, there were very few patients out there that were willing to give me some skin biopsies. And what we wanted to understand is how the genes are regulated in ALS. Because we wanted to understand gene regulation, we wanted to see a mutation that may be related to genes regulations, like TDP43. And the other thing that we were looking for, it's a mutation that is not only present on sporadic cases, but also present in familiar cases. So TDP43 accomplished all of those requisites. It models or it controls the gene expression, and it's present both in sporadic ALS and familiar ALS. So we needed to have a proof of concept, which means that we needed to try at those times whether these stem cells derived neurons will be a bona fide stem cell, a bona fide neuron. And what we tried early on was to generate stem cells from mouse. So in our proof of principle, we took the transgenic ALS mouse that overexpressed that mutation on the superoxide dismutase, and a mouse that expressed HB9-GFP, which is a promoter that gets to be expressed on the motor neuron. So when we cross these two animals, we can actually have motor neurons in green, which is pretty cool, I think. Actually, we are bringing this mice is traveling now. It's going to arrive in 2 weeks. And this one is going to arrive in 1 week. We are going to have it in our university. It has been a huge pain in the neck, super difficult to bring all of these guys. We need to build a whole animal facility to get them, not because they require too many things. They are not immunodepressed mice, but it's very difficult to do science here. And I bet you know, we are all here trying to battle with the same tools. But these little details that will be nothing here are very difficult. And so we cross these animals, and at 3 days of the cross, we take the uterus and isolate the blastocytes, and the blastocytes then are dissociated and cultured over a MEF layer. So what happened here is that I have the MEF monolayer, the mouse embryonic fibroblast, and this is a host for these colonies of cells that come from the blastocysts and that start to form a colony. With this, I can generate different lines that either have the ALS phenotype or are wild type for ALS. But we are not talking about any SOD1. We are talking about the human SOD1, repeated in tandem 18 times. With this, what we want to accomplish is to have a model system that is analogous to the mice system. And you may wonder why. The idea of this is that this mice has the clinical feature of an ALS patient. So in the mouse, you can follow all the clinical features of ALS. And that, people, is really hard to neurodegenerative disease. I bet you have the same problem trying to find a transgenic mice that replicates Alzheimer's, or a transgenic mice that replicate any other disease. They may have the phenotype, they may have partially one or two things about the disease, but they don't replicate almost never the clinical features. This one does, and that makes it a perfect model. So when we did this, we also tried to take these stem cells and differentiate it into motor neurons. And here you can see the protocol. It lasts about 12 days. Here you can see a representative images of what happened when you just seed the cells. Then when you do something that is called embryobodies, which is that you take your stem cells, dissociate them single cell, and then these cells turn to aggregate. And in these aggregates, there are spheres. They can grow in suspension. And the trick is here, because once you have the embryobodies and you have the stem cells in a sphere, you can treat them with factors to reach differentiation. But when you treat them, those factors will touch the cells in a particular way. It will be homogeneous. Imagine you have cells in flat, and you add something on top of it. But then the cells start to grow at layer 1, layer 2, layer 3. It's very difficult for each of these cells homogeneously react to a particular trophic factor. The sphere gives you that advantage. So these embryobodies are very widely used on stem cell biology to try to generate conditions so that you can do a very good differentiation into, let's say, interneuron, hippocampal neuron, or motor neuron. So we differentiate them into motor neurons, and then, because I was in a wire lab, because I was working with Magdalena, we learned very early on that neurons need glia to survive. I bet if something we know about this university is that first, they need glia to survive, and second, we culture neurons not for them to die. We culture neurons to observe them. And this is something very tricky in literature, because usually you see papers out there that show a neuron, 7 days neuron, or 10-day neuron, and experiment done. That is not the idea for neurodegenerative disease. We need to culture a neuron so that it survives, so it matures in culture, and it ages in culture. And that's something we need to figure it out really early on this system. So we culture them in glia. We knew we have the wild type human SOD1, and we have the mutant SOD1 human. We did the monolayers, and we put on top of them the neurons. And I remember I was super excited. I was like, oh, we need to record them. We need to do the calcium imaging. We need to do this, that. And my boss at the time told me, don't do any of that yet. Just count them. It was so incredible. We just count the cells, and we figured out this. So when you culture neurons that either have the ALS or are wild type for the human SOD1 protein or non-transgenic over wild type glia, you have this curve of natural cell death in vitro. This is over a period of 1 week, 2 weeks, 3 weeks, and 4 weeks. We can actually keep them very long in vitro. This is just what happened on that publication, but you can keep them for 8 weeks if you want. It's very, very long. Now, when you culture them with ALS glia, what happens is that survival curve gets to be very, very dramatically affected. And when you culture them, the neurons, the motor neurons that have ALS and the glia that has ALS, you have a cell death that is dramatic. This was the first time somebody published that the ALS or a neurodegenerative disease is not only because the neurons are dying, it's because the astrocytes or the milieu, the niche in which the neurons are growing, it's toxic for them. Of course, after that, we tried for years to find the toxic factor. It's very difficult. Still, we don't have the tools to figure it out what is exactly the factor or the many factors that are killing the neurons. But it was a good start. At the same time, the same year that we were trying to do this, Yamanaka came out with this ability to take biopsies from patients and differentiate it into whatever you want. So we went, this is a summary of what we found on the mouse model. I'm not going to go in detail on this, but this is what happened on 2006 and 7, and now we are already 10 years into the system, and we know more things about it. Of course, we have plenty to know about what we can ask to iPS-derived neurons, but basically what we could do in theory is that you can take a patient, take the fibroblasts, make the IPS, and here you can have two different types of research. You can try or envision to take this cell, repair the cell, repair the mutation, repair the problem, then differentiate into neurons, and then go again to transplant into a human or a patient. Now, with neurodegenerative disease, that's a big problem because it's very difficult to access to those areas of the brain. I will say exception could be Parkinson, but it's very difficult to access to an area of the spinal cord without making major damage on the patient. In 2015, a group of Canadians figured out a surgery that could have hopes on that sense. But that's a major challenge. Okay, we have the cells. Maybe we can fix them with CRISPR or some other technique if we know the mutation, because 90% of ALS cases, we have no clue what's going on. Now, in those cases, you could think about doing a replacement theory, which is taking those fixed cells and putting them back into the person. But then imagine everything goes fine. And you take the cells and they go into the spinal cord. And they magically survive, because that's another issue. Imagine that you are in the best case scenario. Now those neurons need to come out of the spinal cord to target a muscle that is, Fernando, can you come here? Check out this. This guy is huge, see? And so these neurons, no, don't go, you're not. So these neurons are around here. They are usually on the two swellings, the ones that are connected to the upper arms and to the legs here in the lumbar area. This neuron needs to come from here to here. That's, that's difficult, that's challenging. And so I really hate when people talk about these stem cells and promise this will be the cure for everything. If there is one message I want to say today, this is, could be, we are working for it, but right now it's not. And it's terrible because I can see patients all the time, and they are always telling me, I want to pay so-and-so to go to so-so and do something. It doesn't work. It hasn't worked for years. Even the guy who invented this technology, it's still working on clinical trial to try to do replacement at the level of diseases on the eye or diseases on the skin. To try to go on the nervous system right now is, quite frankly, very naive and somehow cruel to the patients. So now what we could do, though, is to take these cells and to use them as an in vitro model to study drugs. And this is really, I think this is really cool. We can do this now. Maybe we will be able to do this later. But right now we can do this. We can try to understand how the neuron die. We can try to understand what are the genes that are associated to the cell death process. We can do a time course where we can see an early ALS neuron, a mature ALS neuron, and an age ALS neuron. The same way we can do it with ALS, we can do it with Parkinson, with Huntington, with Alzheimer's. And actually, now in the lab, we are carrying iPS cells from Huntington patient and from Parkinson patient. And the idea is that you could test on a patient-specific level a particular treatment, which is something that could be very powerful. And right now is leading on the idea of precision medicine. Because what we could do is, if you have nowadays 500,000 drugs, and you think that from those 500,000 drugs, maybe you can target 10,000. This can be automated. I think in Germany they do it in 2 weeks. And you can actually submit your cells and get the data in a period that is reasonable enough for you to think of embarking on some treatment, which is something really powerful. And I think this is going to happen very soon. A little bit more of that in here. You can take the fibroblasts, reprogram them, try to do patient-specific therapy, try to test drugs, and try to see what's going on with your transcriptome. This is a table that shows an example of how many iPS cells have been trying to use to study ALS, spinal muscular atrophy, Parkinson's disease, Huntington, Down syndrome, and the list goes on and on. And here you can see when we have the genotype, the disease phenotype in interest cells, sometimes we see a phenotype, sometimes we don't see a phenotype at all. So it's kind of tricky, and we have to learn with the test and failure. So what we are going to do now is we are going to take one of these ALS IPS line that carries a TDP43 mutation that is very important in ALS because it has been proved to be present in sporadic ALS and familiar ALS. And here we are going to try to see whether these mutations somehow act in the same way in an in vitro model, in a human model in a dish. We have different lines. I'm not going to go in detail with that, but our experimental paradigm is this. We take these embryonic bodies, we differentiate it for 4 weeks, and then after that, we plate the neurons on a cover slip that has 3 dots of paraffin. The reason I'm doing these dots of paraffin is like to make a mechanic tripod. And so then, in another set of experiments, I can culture the glia, and then I culture both cells in close proximity but not in direct contact. And that's really cool because sometimes you want to do biochemistry on the neurons, or you want to do RNA-seq assays. You will have the possibility to do it in a purified population instead of in a mixture. Actually, I did all of the controls thinking, okay, maybe if the neurons don't touch the glia, they could have something like in terms of phenotype that could change, and it doesn't. When the neurons touch the glia, the toxicity is faster, like 20% faster than when the neuron is in close proximity, but it happens anyways. And after that, we can culture these neurons for 4 to 6 weeks and do many different types of experiments. We initially focused on RNA extraction to do RNA-seq experiments, but we also did survival assays, cytoskeleton assays, neuronal transport assays, and a bunch of other stuff. And the idea is that here you can see how this cell look like at 7 days in culture or at 21 days in culture when we look at a control line. This, of course, doesn't have the mutation. Here you can see the cells starting to grow where they start to project their projections. This in green is HP9, a marker for motor neuron. And here at 21 days, we switch to another marker, CHAT, and you can see a neuropila completely formed. One of the first challenges is that not every line has the same efficiency of motor neuron production, which is kind of difficult because you want to start with the same number of cells. So what we had to do is characterize the line, see what is the percentage of motor neuron production, and then isolate the motor neurons to try to start from a clean start. Here is the way we did it, and here I can show you that in these lines that carry the mutation, we saw the beginning of a phenotype with TDP aggregation. Now, TDP, when it's muted on ALS patient, what happened is that it translocates completely from the nucleus to the cytoplasm. Now, this is a protein that is a repressor of transcription. It should be on the nuclei, and in the case of ALS, it goes everything to the cytoplasm. So it's very difficult to understand why a protein that functions in the nuclei goes to the cytoplasm. And the paradigm is huge on the field. It could happen that this protein has a loss of function, meaning that TDP43 is not doing the role that needs to be doing, or that maybe it acquires a function, and we can call it a toxic function, that somehow it's killing the neurons. So, going back to our initial paradigm, we wanted to test this on the same model of the mouse system. And I'm not going to show you this because we already see it, but one of the things that I wanted to show you today is that, so we know that the glia that I used to induce the death of motor neurons had an SOD1 protein that is mutated. And one of the things that we wanted to know is how this SOD1 mutated gets to be aggregated. There is a huge number of studies made on this field, but it's still not clear. It's still, even though now we have the crystal structure of SOD1, we do not understand how the aggregates form early on. We know how they are later on the disease, when they are big, perinuclear, and they are all over those neurons that die. But we don't really know how they do it when in the initial part of the disease. And this is something we look forward to understand. And so we know that SOD1, when it's a normal, it acts as a dimer. But when it's at the form that is mutated, that dimer gets to be dissociated, and you have monomers that can aggregate in big clumps of protein aggregates of SOD1 in the neuron. And here you can see image of that. Here there is a control and an SOD1. Now, what we are doing now is we are trying to model this at the level of aggregation, but doing some technique that is called coarse grain. And what they do is they model a cube, and in this cube, you can actually reduce the amount of information on the protein already folded and try to replicate the aggregation in a silico. Because we want to do a correlation between what could happen in terms of the aggregation in the silico and what happened on the cells in vitro. We have a few candidates, mainly the binding site for copper and zinc. And here, it's our experimental line. You can see that on the mouse experiments, we usually did it in 12 days first of differentiation, then plating, and then 4 weeks of experimentation. In the human case, it's way more difficult because you have to take the cells, make the EV, do the induction for a whole month, and then plate the cells just there to try to wait for 1 to 12 weeks. And why is that? It's because the human-derived neurons don't mature at the same speed that the mouse ones. And so for you to see the phenotype, you need to wait way longer in the period of that neuron. Usually the physiological activities start early. They start a week or 10 days. But in terms of the phenotypic activity of the disease, it's usually after 4 and even after 10 weeks, depending of the line that you are working with. And this is representative images of the first cells produced in Talca. That's why I bring them here, because it's really difficult. It's a 12 weeks experiment in the sandwich. And this you can see at 5, 7, 14, 21, 28. And we have even now, I think we are going into the 38 days or something like that. So it is difficult. I have a tremendous group of students that, you know, for working with these guys, you have to work every day. It's not like they have a Saturday and Sunday off. You need to go to the lab a Saturday and a Sunday, holiday, Christmas, you continue. And so it's really difficult to work with these cells. And the other thing that happened is that they grow without antibiotics. So we had to work a lot before even bringing the cells to be able to culture them for 12 weeks without a single antibiotic in the dish. And so it's tricky, but it's possible. If we are making it in Talca, I think it's possible to make anything because it has been quite a challenge. And the paradigm that we are going to use now is to take these human-derived motor neurons and culture them with non-transgenic glia, wild-type glia, and mutant glia. And then we want to observe these mutant neurons stress, because I don't want my grad student to wait 12 weeks for a phenotype. Actually, I'm using it, the glia that I know kills the neuron, I'm using it as a stress model. Because asking somebody 12 weeks of their life is a lot. So we are going to accelerate the phenotype of the disease, the appearance of the disease, by stressing them with the glia. And here you can see representative images of how the cell looks like at 2 weeks when you have control neurons over control glia, and here when you have mutant neurons over mutant glia. So the difference is striking. There is a survival issue, there is changes in the neuropila, there is fragmentation. We have seen aggregations of all sorts, and what we are doing is, this is the survival curve, it's a 30% of survival death, 40% at 14 days, and it's even bigger when you go 14 to 21 days. And here you can see a representative image of the neuropila. You can see obviously less cells at 2 weeks. And now we are starting to see the gene patterns on these cells. Here you can have a triplicate, a technical triplicate of these cells when you have the mutant line or when you have the wild type line. So we can see that at least they differ from each other, but the pattern is pretty similar between these and pretty similar between these. And we can ask now, what are the genes that are upregulated and what are the genes that are downregulated? Moreover, we can do gene ontology and try to understand what's going on with those genes that are enriched in the neurons that are from ALS patients. We know that they are enriched in nervous system development, in ion channel activity, metabotropic glutamate receptors. And when we ask about what they have downregulated, we can see that every group or the major groups are associated to the way the cells communicate between each other. Those proteins that are associated to cell communication are usually downregulated on ALS, on these ALS-derived neurons. We can start asking questions about why they die because we have this information, and we want to combine the information of the transcriptome with the cell biology. We don't want to take them apart, we just want to combine them to try to see, for example, I know that in my mutant line I have an increase on ion channel, an increase on neurotransmitter receptor, and obviously this could lead to abnormal excitotoxicity, which could trigger death. So now we have all the tools to try to prove this hypothesis because we have the transcriptome data, we can do electrophysiology on these cells, and we can see whether that's the case, or even better, if by blocking excitotoxicity, we may decrease cell death in our system. There are other ideas. For example, these proteins that are ADAMs are secreted. We know that the glia plays a huge role, so we think that maybe we know that our mutant lines decrease the expression of ADAM, but increase the expression in glia, and that could lead into cell death too. We have a lot of ideas of what could be going on the cells, and now we want to do and take one of these ideas and prove them, correlate them, the transcriptome and the cell biology. And this is what we are doing now in the lab. We have successfully prepare RNA samples from motor neuron from patients. We have many hundreds of genes that are consistently up or down in, of course, 3 different biological replicates, which is really difficult because you have to start with 500,000 cells per well, and you usually do a 24 well per experiment that should be triplicated for a technical replicate, so that's a lot of cells. And we know that by analyzing the transcriptome, that there is a differential splicing in these cells. These are the lines that are currently on the lab. We have control, we have ALS, Huntington, and cerebral palsy, and we are getting Parkinson too. And these are the lines that are currently available in certain consortiums in New Jersey and in New York. So basically, nowadays, you can actually take an agreement with a consortium and say, I'm interested in this particular pathway, and I would like to use your cell line that comes from a patient to study that pathway. And you can actually get them. And it's not so expensive. The issue is to bring them to Chile. That's another issue. We can talk like 3 hours on that topic, but not now. I know that you are hungry. And that's mainly all that I wanted to talk to you. This is my experimental model. We want to see survival, ALS hallmarks, gene expression, and function of IMNs in a model that is human. Oops. I don't know what happened. Here. Here, the lab, we are very few still, but we have a postdoc, Alejandro Maureira, from here, so probably you know him. I have a grad student, Daniela Cáceres. She and Carlos are doing all those 12-week-long experiments. And of course, my Juan Carlos that you all know, that helps a lot. Thank you, and I'll take any questions.
Si hay alguna pregunta.
Did you record this cells?
Yeah.
Yeah, and what happened? They present synaptic activity?
They have, they start, the beginning of the synaptic activity is about 10 days. But the changes really happen in 10 days. And the changes are associated to AMPA activity.
Look like a normal neuron.
Yeah, well, kind of. Not. They have a lot of capacitance. They are huge. They are very big. Capacitance is high, and they have a lot of activity for, like, AMPA type.
And do you see some difference between the wild type cells and the
Increase on frequency and increase of amplitude of the AMPA activity in the ALS cells.
Sí.
So, technical question. So, you take a sample of skin and then you transform these cells to motor neurons. So, and you induce the expression of some genes. So,
Yeah.
how you do that?
With a kit, you buy it. Pretty good. There are several companies that for $3,000 they give you a kit, and you just have the whole virus particle already packaged, so you treat the cells. The difficult part is not that. The difficult part is to selecting the clone that is actually a good clone. Because once the cells are reprogrammed to be pluripotent, they generate, let's say, easily 1,000 clones on the ditch. You have to pick them up by hand, and then you need to test them, whether they are good cells, whether they have a good karyotype, whether they can actually make a tumor or they can generate the 3-layer of tissues from that particular clone, and that's called the teratoma assay. Or you can simply take them in an empty dish, let them differentiate alone, and look for the 3 different markers. So there is a lot of quality control on these cells. You have to test for a bunch of pluripotent markers, and that usually takes anything between 3 months and a year. Just the characterization. And then
But
after that, you're fine.
You treat with factors only? Or
You could do it several ways. One is virus. There are several viruses that have been used to do this, but people figured out pretty early on that if you do it with virus, then you have a constant expression of c-MYC, and that's not good because you can induce a cancer in a patient that if you want to think about replacement therapy, you can actually induce a cancer forming, and this is what happened on the first clinical trial, I think. And the other thing that you could do is RNA, directly add the RNA and try the differentiation, and lately what you can do is actually add the proteins. So usually the people who are trying to do this are in New York and Japan, and they are being very successful. Every day they are getting better at doing this reprogramming without messing up with the cells. So it's hopeful.
So I will ask Magdalena here to give her this diploma for her participation. Thank you very much, Natal. Mónica, Nat.